Okay, it's a pleasure to follow Darryl. We've been doing this team presentation for a couple of days now. I'm going to skip a few slides here. You're doing non-invasive prenatal testing on cell-free plasma DNA here, so you're familiar with that process. It's tremendously effective. It's quite expensive. It requires an infrastructure for handling the data and doing the sequencing that's awesome. And in pregnancy, the good news is you only have to do it once per patient. What we're going to be talking about for the rest of this symposium is trying to transition that kind of technology to cancer. And the problem with cancer is you can't just do it once per patient because, as Darryl showed you in many examples, the cancer is an evolving disease in each patient, and you have to keep watching what's happening. Okay, so I think it's pretty clear that it's useful to do NGS as a discovery tool with cancer patients, both for copy numbers and for somatic mutations. But it needs a backup for routine testing to monitor what's happening when you're treating the patient. Okay, and that's where we think this nucleic acid mass spectrometry is probably the most cost-effective solution, and it certainly seems to work very well thus far. So let me start by showing you another solid biopsy. This happens to be an EGFR mutation. I don't know which one in a lung cancer sample. Once again, an FFPE sample. And just as in the example Darryl showed earlier in colon cancer, the wild-type allele usually presents as quite a strong signal. And the mutant allele, which is the actionable allele for the choice of drug, is weak. Why is the mutant allele weak? There are two reasons. In any actual solid biopsy, typically most of the cells are normal. They can be stromal cells. Increasingly, we're realizing that if your patient immune system is fighting the tumor, they're immune cells, okay? And they don't have the somatic mutation, okay? So that's why it's fractional. The second reason is that tumors are heterogeneous. Not all of the tumor cells have the same somatic mutations. And depending on where the biopsy was taken in the tumor, as I'm about to show you, you can get totally different results. Okay? So this is really a problem because not many patients are going to let you take 10 different biopsies. And even if you could afford to sample a tumor in many places, you would then, the cost of then doing that kind of an assay could become prohibitive. So to look at this, we've been collaborating with Charlie Swanton in London, who has managed to get approved protocols and compliant patients to undergo multiple biopsies. And what I'm going to show you is lung cancer again, where the patients have at least 2 primary biopsies and at least 1 metastasis. And what Swanton does is to take these solid biopsies and do whole exome sequencing very deeply, and that takes 4 to 5 weeks by the time the analysis is completed. It's one of the problems with NGS in cancer. It's not a lookup table like prenatal. It requires a lot of analysis. And then he gives us the data file after he's figured out which are the false positives and which are the true positives. And typically there are 5 to 20 or 30 unique or common mutations in that patient. And we design a custom mass array assay to look for those mutations in the same patient sample. And it takes us 3 to 5 working days to design the assay and run it, okay? So you can see how much faster this is, and of course it's tremendously cheaper. When this was done on 6 patients, I know it's a small number, you can see that the number of potentially clinically actionable mutations is large, spread among 3 or 4 different biopsies. But what's very depressing is if you ask, well, how many of these actionable mutations are present in all the biopsies? And therefore, how many of these patients have potentially, can potentially be treated with a monotherapy? You can see that for some patients, most of the mutations are just not present in all of the current tumor growths. And in fact, many, actually most, of the mutations are present only in a single biopsy.
Sorry. Just one clarification.
Sure.
You mean the mutation or the gene?
Well, the gene presumably is in the genome and is present in every cell unless, it can tolerate a deletion. The mutations.
Because it doesn't matter if there's one EGFR mutation or the other activating EGFR, but it's in the same gene, empirically the treatment is the same.
Right. Actually, I happen to know the details of all this data, and there's no case here where there are 2 different mutations in the same gene. Okay? So in this particular case, I can't speak in general, but in these patients, it's actually then between 7 and 10 different genes. Okay? So this is a very, very difficult situation, and I think most people agree that by the time a patient gets to this stage where they have 3 or 4 different diseases, you're not going to cure them, right? You're not going to save them. It turns out that the concordance between next-generation sequencing and DNA mass spectrometry is close to 100%, and usually any differences can be explained either because the assay for a particular mutation wasn't present in the multiplex for the mass spectrometry, or because the sequencing wasn't sensitive enough to see a low-level mutation that was picked up. Okay? So what this says is that if we're doing solid biopsy, we have a almost certain probability, unless it's just a single focus for the tumor, of missing somatic mutations that may be actionable. Okay? Never mind the cost and the unpleasantness of doing that solid biopsy. It's just not sampling the tumor properly. Okay? So what do we do? Well, if we look in plasma, instead of looking at a sample of a tumor, in principle, we might sample all the versions of that tumor. And that's what I'm going to show you. And of course, measurements in plasma are non-invasive. We can repeat them as often as we like. If we're using mass spectrometry, they're inexpensive. And currently, most people are focused on starting with that solid biopsy and then building confirmatory panels and monitoring panels for liquid biopsy. Right? But I think you can make a very strong case for starting with the invasive biopsy. Starting with the non-invasive biopsy, starting with the liquid biopsy instead of doing surgery on the patient to take a solid biopsy. All right? Because if you see an actionable mutation, you can start to treat. And then I'll show you in a few minutes, you can then use the liquid biopsy to monitor if the treatment is working. Okay? And you haven't had to do surgery on the patient. So this may or may not become a standard of care. I think you probably know, Darryl already mentioned reimbursement. In the U.S., there is no reimbursement for liquid biopsy yet, so it's all research. At the end of this, I'm going to show you that there may be some chances to actually improve the sensitivity of liquid biopsies, even to the point where we might be able to do pre-symptomatic detection. And of course, that would be a dream, because then you'd have a yearly blood test to see if you were developing any cancer. All right, let's spend a couple of minutes understanding the properties of cell-free DNA in plasma. It comes from apoptosis, and it has a half-life of 15 minutes. Okay? So that means that when you see free DNA in plasma, you're looking at DNA from cells that have died within the last 15 to 30 minutes. Okay? So it's a very dynamic measurement. When you're looking at a biopsy of a tumor, you don't know if the cells are alive or dead. You're just looking at all the DNA in that sample. But that's not the situation in blood. Okay? The problem we have in doing, and Darryl's already talked about this, doing cell-free DNA measurements in plasma is that the overwhelming majority of DNA in plasma is cellular. So if you break any of the cells, you've ruined the sample. Okay? And Darryl showed you one way to use QC to check to see if you've broken any of the cells. Now, of on average, about 1,000 molecules per cc of any genome sequence in plasma in a pregnant woman or a cancer patient or anything else, about 90% of them come from apoptotic white blood cells and apoptotic vasculature. Okay? And that's not interesting because that's wild type. Okay? So only about 10% typically of DNA comes from remote sites. This is sure in pregnancy, and it's sure in early cancer, although in very late cancer with huge tumor burdens, then you can get much more. Okay? So if we have a tumor which is 10% EGFR mutant, and we look for that allele in plasma, it's going to be 1/10 of 10% or 1%. Okay? So the methods that Darryl has showed you up till now with about a 5% sensitivity limit simply can't be used in plasma. They might work for some patients, but they won't work for all patients. Okay? The DNA in plasma is small. Darryl's already mentioned that. And just in case you're wondering, lots of people are promoting using endosomes or circulating tumor cells instead of using cell-free plasma DNA. I think that's a completely expensive waste of money and time because we barely have enough cell-free DNA molecules to work with. And if you go to endosomes or tumor cells, you reduce the number of available molecules by a factor of 10 or 100 on average, and that's just not enough to work with. Okay?
So why is the difference in 160 and 125?
That I wasn't going to talk about, but since you mentioned it, I will. Okay? If you ask a nucleic acid molecular biologist what that difference means, it's nucleosomes plus or minus histone H1. Okay? If you ask whether anybody has done the experiment to prove that there is histone on this DNA and that H1 is either present or absent, no one has ever done that experiment. And the fact that no one has ever done that experiment is a crime, but it shows you that once people learn to work with nucleic acids, it's so easy that they lose the technologies that would be required to show histones on those nucleosomes. You know, we don't have nucleic acids. We can start with one molecule, as you'll see momentarily. With protein, you need a lot of material, and it's hard to do it. So we don't know. Okay? And this has been seen in 3 or 4 different clinical presentations. So it's a general phenomenon, but nobody has an explanation for it.
So that could be a very good marker to quantitate your...
Unfortunately, although the mean size shifts, the distribution is broad. Okay? And Dennis Lowe has actually used that size difference as a discriminator in prenatal. And it works well, but to use it, you have to do paired-end sequencing instead of single-end sequencing, and so it's much more expensive. Okay? But it is a useful marker. So in the past 2 years, there have been about 20 publications with very small numbers of patients showing that if you monitor the right tumor-specific sequence in plasma, you get information that's useful. And I just picked this one example. It's a woman with ovarian cancer, and various techniques were used to look at various different somatic mutations which were peculiar to the tumor. And what you can see is one of these was extremely informative. Whichever this one is, a woman was put on that therapy, whatever ECX is. She responded very well. The plasma level of that mutant sequence dropped precipitously, but eventually the tumor came back. She was then treated with paclitaxel. Again, there was some response, remission, and so on. Okay, so it's very useful because all these measurements are relatively inexpensive and they're non-invasive. But if you think about the biology of what should be happening here, this is not the right experiment. Okay? If I have a drug that's effective against tumor cells, and I give the patient that drug, I should see a huge spike of mutant sequence in the plasma, okay? Because I've killed those cells. And that's, in fact, what you do see if you sample at short enough times. And these are some results from Trobogene on a patient who has this famous EGFR T790M resistant lung cancer. And this patient's being treated with the new experimental drug that is active against these resistance mutations. And here we're looking at the mutation itself in plasma or an exon 19 deletion in plasma, both of which represent drug-sensitive tumor cells. And you can see these are, this is 12 weeks, okay? So each of this patient was analyzed every week, and you can see that in the first week, everything happens. Okay? The drug is tremendously effective. It kills what appears to be almost all of the drug-sensitive clones. And then maybe there's a little bit more killing. Maybe there's a second population that is killed a few weeks later. If you use imaging on this patient instead of molecular analysis, you don't see anything. Right? So this, I mean, we have so little data, but I think this is clearly the right experiment to do. People will do this now in lots of situations, and it should allow, I think, very intelligent clinical decisions to be made. Because you have to ask yourself, at this point, when I'm essentially at baseline, why am I continuing to give this patient an expensive drug? It's done its work. Okay? I ought to stop. All I'm doing at this point is selecting resistant clones. Okay? If I continue to monitor the patient and the mutation comes back, then I should use the drug again. Okay? So this kind of drug holiday, drug alternation therapy, which is right now totally experimental and just being done in a few places, looks very promising. And it's made possible by the ability, in this case it happens to be urine rather than blood, but by the ability to do sort of continual monitoring of patients. Okay? But to do this kind of monitoring, we need very sensitive measurements. Okay? And this is the trick that we use to pump up the sensitivity of the DNA mass spectrometry so that we can see very low levels of mutant sequences. So we're not interested in the wild type allele. We know it already. So why bother to detect it? What we do is leave out the polymerase substrate that allows a primer to be extended on the wild type template. So we eliminate that extension product altogether, and we cycle the sequencing reaction until in one cycle a primer sits down on the rare mutant template, however rare it is, and then it's extended by a single base and stops. Okay? So I think you can understand that with a trick like this, we can force the detection of any molecules of mutant template, okay? So long as the polymerase doesn't misincorporate. So if you look just at the red arrows, this shows an assay for a particular, in a spike-in experiment, a particular mutant sequence. This is a non-templated control, so there's no peak. This is a wild-type template, so there's no peak. This is a spike-in at 0.5% mutant sequence, and you can see not only is there a peak, but it's a very, very strong peak. Okay? That's because we're forcing the detection. Okay, and we actually have a commercial panel of 26 mutations that you can use off the shelf, or we have design software that allows you to design your own multiplexers. And the way this panel works, it's a single PCR reaction on 40 nanograms of DNA, and then it's split into 4 primer extension reactions and 4 different sequencing reactions. So it uses 4 tubes, 4 wells in the mass spectrometer. All right, I'm going to show you briefly a pilot project we did together with a large pharma looking at a set of patients with the famous V600E mutation in BRAF in melanoma. We had a reasonable number of patients, controls, and what I'm plotting is the intensity of that mutant peak for all the patients versus negative controls and spike in positive controls. And this is a 0.5% and a 1% allele frequency spike in positive. So if I blow up just this region of the plot, now I have my patient samples. This is 1% allele frequency. So all of these patients have less than 1% allele frequency. Those are my negative controls. So all of this signal is real. And some of these patients, I estimate from this data, we are detecting only a single mutant molecule. Okay? So you simply can't get more sensitive than this. Okay? And this is, I'm sure you know, this is a clinically actionable mutation. So these patients with very low levels should be getting drug. Now, what's of interest, and somewhat confusing at first, is to compare liquid biopsy with solid biopsy. And we have that for these patients. Forget this PCR result. Let's just look at the solid biopsy in yellow and the liquid biopsy in blue. In this case, there's about 75% concordance. But in this case, in the liquid biopsy, we miss about a quarter of the patients. And we see one patient that's not present in the tumor. All right? But in other studies, it's the other way around. You can have a lot of false negatives. You can have a lot of things that, if you didn't know better, you would call false positives. So just remember what's going on here. The tumor is heterogeneous. So depending on where you sample, you may or may not see mutations. The plasma is seeing the tumor cells that have just died, and nobody knows at this point how well they represent the entirety of the tumor. And in addition, because we have so few molecules, as Darryl has already mentioned, we will have false negatives because some patients don't have enough molecules to see. All right? So here's another set of data from Trovagene. Trovagene is interested in urine. I think at Agena we currently prefer plasma, although we could do urine. And what's being plotted here is KRAS copies, tumor-specific mutant sequence per sample in urine, which is probably about a liter, and in plasma, which is probably a few cc's. Okay? And you can see that there are many more tumor-specific molecules in that urine sample. Of course, it's also a much bigger sample. But what I want you to notice is that the range, the number of molecules of mutant sequence in patients in either urine or plasma varies by 2 orders of magnitude. Now, I'm sure you've seen exactly the same thing here with pregnant women. Okay. And nobody knows why some people have high plasma levels of circulating nucleic acids, regardless of the clinical situation, and other people have very low levels. Okay? We just don't know. But the problem is, if we're going to design robust diagnostic tests, we have to either have controls to rule out false negatives, or we have to accept the fact that they're going to be false negatives due to stochastic noise, due to sampling noise. And now let's drill down a little bit on the issue of stochastic noise. The moment you have less than a few hundred molecules of analyte, you lose your ability to quantify well, and you begin to have a significant risk of missing a minor allele altogether. And sampling noise scales as the square root of the number of molecules. Okay? So if you have 100 molecules, you're going to have 10% noise, so nothing will be measurable with an accuracy more than plus or minus 10%. And if you have 10 molecules, you have 30% noise, plus or minus 3. Okay? So this is a problem in fundamental physics. Okay? If you ask a physicist how to overcome stochastic noise, he or she will say, well, it's easy. Repeat the measurement 100 times. But you know our measurements, your measurements on plasma, are consuming the whole 10 cc blood draw. So you can't repeat it 100 times. All right? So what do? The trick is, instead of trying to average across replica samples, we can average within one sample across replica markers. And that's what makes NIPT work. because you're not just using one chromosome 21 specific sequence in the Natera test, you're using a huge number, okay? And instead, at Sequenome, where we're counting molecules instead of looking at alleles, it's the same situation. Any read we get from chromosome 21 is useful information. It doesn't matter where on the chromosome we're reading, okay? So that's the notion. And I want to show you in the last few minutes two very recent approaches that we're taking to try to improve the situation. Okay? I mean, it's a fundamental problem, but there are some ways to think about it intelligently and try to up the sensitivity, which will help then for those patients who adventitiously have very small numbers of molecules. And the first of these is a trick which we call sensitivity enhancement by fragmentation. And rather than read this slide, it's easier if I show you a picture, and I'll show that to you in the context of digital PCR, but it's the same phenomenon for any detection system. It's just easiest to understand it, I think, by digital PCR. If I have one target molecule, and I look for it in a digital PCR assay, I'm going to get one positive well, and all the other wells are going to be negative, okay? That's my molecule. Well, but if I have stochastic noise, I may not have the molecule. Okay? The way to improve signal to noise is to take the molecule and cut it up into lots of pieces and design a multiplex assay which goes for any or all of the pieces. Okay? Now, instead of having one molecule, de facto, I have many molecules. And so my assay sensitivity goes up. It's that straightforward. You can't do this with real-time PCR because you can't multiplex. But you can do it with the mass array system. And here's an example. It's showing today. Okay, so on the left are single marker assays. These are factors of 2 dilutions. And this is about 1 molecule per sample. Okay, so this is down in the limit of infinite dilution. And here the red and the black are scoring single analytes, but the green is scoring 1 of 2, and you can see it's more sensitive. Here this blue is scoring 1 of 4, and you can see it's much more sensitive. And here the gray is scoring 1 of 7, and it's much more sensitive still. Okay? So by using multiplex assay and by fragmenting my target into as small a pieces as my detection allows, I can gain, in this case, more than a log in sensitivity. I can actually detect, on average, less than 1 molecule. Okay? Because, in fact, I've blown it up to be many.
This is in genome DNA?
Excuse me?
This is done in genome DNA?
This is actually done on human genomic DNA, yeah. These are actually nuclear markers in a complex assay I'm about to show you, yeah. Okay, the other way to do this, just look at the bottom, is to find repeated sequences that are diagnostic, okay? Because then I don't have to fragment them, then I have multiple copies, okay? So we become very interested in thinking about where in the human genome can we find highly repeated sequences which have diagnostic value. And what has caught our attention and what we're now very actively engaged in is trying to use mitochondrial DNA. Now, in an average cell, there's about 1,000 copies of the mitochondrial genome. Okay? So roughly 500 times more DNA than any single copy locus on the nuclear genome. Okay? And we were curious to see how stable the ratio of mitochondrial DNA to nuclear DNA is in normal people. Okay? As a prelude, it is known in all kinds of disease situations that if you look in plasma or in tissue, you get abnormal ratios of mitochondria to nuclear DNA. So in cancer, mitochondrial DNA is very, very abundant. Because tumors are using up a lot of energy, so the tumor cells or the cells in the nearby tissue have to have a lot of mitochondria. In neurodegenerative disease, many of those major diseases like Alzheimer's and Parkinson's, there's a lot of mitochondrial death, which may even be driving the disease. Okay, and so those cells in those under those situations have very few mitochondria, relatively speaking. Okay, but if we want to have a useful assay for any of those disease situations, we have to have a stable background in healthy people, and that's what I'm going to show you momentarily. Okay, so we actually have a 20-plex assay that samples all around the 16,000 base pair mitochondrial genome. And what we're actually doing is each of these assays is a mitochondrial nuclear parallel. That is, it's a sequence which is identical in the mitochondrial genome and in the nuclear genome, except for one base. So we can do a PCR that co-amplifies both genomes, and then a sequencing reaction that discriminates the two. So in this first sort of experiment, we looked at 19 different people. We had plasma from all of them, standard plasma DNA preparation. We did 4 technical replicates on each person, and we're going to look at 10 different mitochondrial assays, and I'm going to average them all together. Okay? So I've got 40 measurements on each person. And what I'm going to plot is the ratio of mitochondrial to nuclear DNA normalized to 1. It's actually around 1,000, but I've normalized it to 1 here, across 19 people. And you can see that this is amazingly tight for most people. Remember, if we look at the actual amount of DNA, I showed you that a couple of slides ago, it ranges by a factor of 100 from person to person. Okay? So it's dancing around all over the place, but the ratio of mitochondrial to nuclear is staying very tight. And people are walking around in reasonably good health with duplications of part of their mitochondrial genome, and that's probably what's happened to this person, or deletions of part of their mitochondrial genome, and that's probably what's happened to this person, because mitochondria are not, they don't have a homogeneous genome. There's this phenomenon called mitochondrial heteroplasmy. You have mixtures of different mitochondrial sequences. So we think this is going to be one very useful diagnostic marker, because it's 1,000 times more abundant than a nuclear marker. Okay, and I think by extension, other families of repeated sequences, which are somehow involved in disease phenotypes, are going to turn out to let us be more sensitive. So with a marker like this, if we could correlate it to eventual occurrence of cancer, it may be that a yearly mitochondrial to DNA ratio test would signal the onset of cancer, because if your mitochondria start to climb up in plasma, something is happening. Okay? So at that point, I'm going to stop, and I'd be glad also to take some questions. Thank you. Yes, please.